Good morning, one and all. Myself, Savinaya K, pursuing second MSc Microbiology from Central University of Tamilnadu. So today I'm going to talk about the topic that is microbial endosymbiosis and epiphytes. So starting with microbial endosymbiosis, it is a phenomenon where one of the species is taken into the cytoplasm of the another species, that means engulfed into the cytoplasm of the another species, that will result in a genome fusion. So it is a kind of relationship where bacteria act as an endosymbiont and multicellular eukaryotic partners, they are the host organism. So endosymbionts are the organism that form a symbiotic relationship with other cell or other organism. We have examples like nitrogen fixing bacteria, that is rhizobia, that is living in the root nodules of the legumes, and single cell algae in the reef building corals, like bacterial endosymbionts that is present inside the insect, etc. So when we talk about the microbial endosymbiosis, with the important thing that comes to our mind is the endosymbiotic theory. So the endosymbiotic theory, it is a scientific theory that proposes that the organelles in the eukaryotic cells such as mitochondria and chloroplasts, they have been originated from a free living prokaryotes like bacteria and archaea. So eventually, like endosymbiosis is as I already said you, it is a relationship between two organisms in which lives within the other organism, and eventually there is a benefit for both of the partners. That is, in other words, we can say it is a mutualistic relationship. In this picture, we can see there is a prokaryotic cell that have cell membrane, nucleoid, cytoplasm. And according to this theory, this prokaryotic cell will grow in size and it will develop infoldings, that is cell membrane infoldings. Further, these cell membrane infoldings will pinch off from the cell membrane and they form a endomembrane system with endoplasmic reticulum that contains endoplasmic reticulum, nuclear membrane, and the membrane bound nucleus. Now this particular cell is considered as a first eukaryote. Now this eukaryotic cell will engulf an aerobic prokaryote, that is proteobacteria. The orange color organism we are seeing in this picture, that is a proteobacterium. So the eukaryotic cell will engulf in proteobacterium. This proteobacterium actually avoid the digestion by the host. That means it is forming an endosymbiotic relationship with the host cell, that is a eukaryotic cell. This proteobacterium has the ability to fix oxygen, that is it can utilize oxygen and it produces an energy which is actually a benefit to the host organism. Now so that is evolved. So then this proteobacterium has evolved as a mitochondria, which is we'll call as a powerhouse of the cell. You can see it is an ancestral heterotrophic eukaryotic animal cell. Coming back to the proteobacteria, first eukaryotic cell, again the cell will engulf a cyanobacteria. The green color organism we are seeing in this picture, that is a cyanobacteria. The cell will engulf a cyanobacteria which manages to avoid the digestion by the host, therefore forming an endosymbiotic association as a primary endosymbiont. So this cyanobacteria actually have a capacity to trap sunlight and perform photosynthesis. That means it can convert the light energy or the solar energy into chemical energy, which is actually a benefit to the host cell. Now this bacteria, this cyanobacteria, later is then evolved into chloroplast. As you can see that that is an ancestral photosynthetic eukaryote plant cell. According to the endosymbiotic theory, the symbiotic origin of the eukaryotic cell is a multi-event process. So there are two types of endosymbiosis, primary endosymbiosis and secondary endosymbiosis. In primary endosymbiosis, it refers to the initial engulfment of the free-living bacterium by the host cell, therefore creating a new organelles within the host cell. The most important and one of the most prominent examples of the primary endosymbiosis are the origin of mitochondria and the origin of chloroplast, in which both were once free living cells. Coming to the origin of mitochondria, a eukaryotic cell engulfed a bacterium that capable of aerobic respiration. Here the bacterium is a proteobacterium. So this bacteria provided a valuable energy source through respiration. So over time, the host cell and the engulfed bacterium, they developed a mutual beneficial relationship between the both. This bacteria later evolved into a mitochondria, retaining its own DNA and membrane structures while working with a host cell. Coming to the origin of chloroplast, here an ancestral host cell that engulfed or that captured a photosynthetic bacterium. Here photosynthetic bacterium is a cyanobacterium which drives the photosynthetic process using sunlight. That means it converts the light energy or the solar energy into the chemical energy and drives the process of photosynthetic process. And this photosynthetic bacterium, that is a cyanobacterium, has evolved into a chloroplast eventually, where it allowed the host cell to use the power of photosynthesis for the energy production. Now coming to the secondary endosymbiosis, it involves a eukaryotic cell engulfing another eukaryotic cell that has already undergone the primary endosymbiosis. Here the main thing is that in secondary endosymbiosis, this secondary engulfment actually results in more complex cellular arrangement, leading to the diversification of eukaryotic lineages and emergence of new types of the cell organelles. Because as we know, the eukaryotic cell is already a complex cell. When this complex cell is engulfed by another complex cell, the complexity increases. Therefore, there will be a more complex cellular arrangement and there will be a more diversity. And we can see new types of organelles will be formed. In secondary endosymbiosis, there is a formation of plastids. So these plastids are the organelles that are involved in the photosynthetic process, and they are found in various algae and various plant species. So different groups of algae have acquired plastids through secondary endosymbiosis, which consists of the engulfment of photosynthetic eukaryotic cell. The next 2 point what we are seeing will be explained in the next slide in more clear cut. In this picture, we can see the clear-cut differentiation between the primary and the secondary endosymbiosis. Initially, there is a eukaryotic cell that contains mitochondria, nucleus, and cell membrane, in which the mitochondria has been evolved from the proteobacteria. When this eukaryotic cell engulfed a free-living cyanobacterium, there is a formation of the primary endosymbiosis, in which it is later evolved into a chloroplast which contain two membranes. Now, when this cell is engulfed by the another eukaryotic host cell, it is called as a secondary endosymbiosis, which consists in which the chloroplast consists of the four membranes. Here, in contrast to the two membranes of the in the primary endosymbiosis, there is a four membrane surrounding the chloroplast that is obtained by the secondary endosymbiosis. And we can see another thing, that is a nucleum, that is a degenerated nucleus here. In most of the cases, the nucleus of the engulfed cell disappears, with the remains of this nucleus still will be found lying between the two pairs of the membranes. So that structure is called as a nucleomorph. So thus we can say that endosymbiotic theory explains the presence of even the double membrane organelles within the protist. So we can see there are some of the examples of the microbial endosymbionts. The first one, in the first picture, we can see the Bushnea aphidicola. It's an obligate symbiont of the aphids, and it is one of the most studied model. In this, it has been shown that this particular bacteria, that is the Bushnea aphidicola, it fulfills the nutritional requirement of the insect host, that is the aphid. And in the second picture, there is a Olbachia, a microorganism that is forming a endosymbiotic relationship with the mosquitoes Aedes aegypti, which confers some of the requirements of the insect host. And the last picture, there is a symbiotic relationship between the sap feeding insect, that is Megacopter cibraria, and its primary bacterial symbiont, that is Candidatus ishikaviella capsulate. And this particular microorganism is actually essential for the host survival during its adulthood. Next is the evidences that support the endosymbiotic theory. So one of the most important evidence is the independent DNA that is found in mitochondria and the chloroplast that supported the theory the most. Other evidences are structural similarities. Mitochondria and chloroplast, they share a similar structure with the free-living bacteria such as double membranes and the DNA. And these both organelles, they almost have a same size as that of the bacterial cell. Now reproduction, mitochondria and chloroplast, they replicate within the cell independently, similar to how the bacteria will reproduce. Genetic evidence, the DNA that is present within the mitochondria and chloroplast, they share more similarity to bacterial DNA than that of the host cell nucleus. And the evolutionary relationship. Analysis of the genetic sequences shows that mitochondria and chloroplast are more closely related to the specific groups of bacteria than the eukaryotic cell. Now come to the importance of the endosymbiotic theory. This theory is important because the theory explains the origin of eukaryotic cell. It also describes how the chloroplast and the mitochondria have been originated from once free-living prokaryotes. This understanding has actually reshaped our perception, like how the fundamental cellular organisms came to be and how the multicellular life forms arose. And last point is a cellular evolution. This endosymbiotic theory also highlights how the cooperation and symbiosis have played a pivotal role in shaping cellular evolution. In next, I'll be playing one of the beautiful video in which you will get a clear-cut idea about the endosymbiotic theory.
There are a lot of amazing theories in science. The general theory of relativity, atomic theory, the cell theory, and one thing we want to point out about a scientific theory is that it's a lot different from how you might use the word theory in your daily life. A scientific theory is not an educated guess. A scientific theory is an explanation of a scientific event, supported by scientific evidence. It must be testable and tested over and over and over again. And while theories can be changed or even disproven, you do want to realize there are a lot of facts behind them. One of our favorite theories of all time is the endosymbiotic theory. We love it because we just find the events in this theory to be amazing. The theory gives an explanation for how eukaryote cells could have evolved from prokaryote cells that lived in symbiosis. Prokaryote symbiosis means these organisms live together. So just to recap from our Intro to Cells video, when you think of prokaryote, think of pro rhyming with no nucleus, no membrane-bound organelles. Eukaryotes, on the other hand, remember eu rhymes with do, and they do have a nucleus, and other membrane-bound organelles. Both prokaryotes and eukaryotes are cells, though, and therefore they do have a few things in common that cells have, including a cell membrane, cytoplasm, ribosomes, and genetic material. But just look at these two cell types and you will see that one is way more simple. So what explanation does the endosymbiotic theory provide for how eukaryote cells could have evolved from prokaryote cells? A long time ago, seriously a long time, over 2 billion years ago, there were prokaryotes, much like there are today. They've been around for a long time. Well, just as there are in many organisms, there was variety in prokaryotes. Some of the prokaryotes had photosynthetic abilities, which means they could make their own food using sunlight energy. And those were the photosynthetic bacteria. Some of these prokaryotes were bacteria that had the ability to use oxygen to produce ATP energy. And some prokaryotes were larger. They could consume others, engulfing them. So obviously they had to be larger. The endosymbiotic theory is that some of these large cells engulfed some of these small bacteria. But instead of those small bacteria getting digested, some of them remained intact, and they actually began to live as symbionts. That means they lived together. This is believed to be the ancestor heterotroph eukaryote cell. And eventually, some of these cells engulfed the small bacteria that could do photosynthesis in addition. And there were some more living as symbionts. This is believed to be the ancestor autotroph eukaryote. Now, it's time to do a little reveal. We consider the bacteria that used oxygen to produce their own energy to have evolved into what is now mitochondria. And the bacteria that use sunlight energy to produce their own food, well, we consider those to have evolved into what are now chloroplasts. It's likely that these ancient eukaryotes had some kind of advantage in their endosymbiosis, and it's also likely that the mitochondria developed first, before the chloroplasts, because if you remember from our Intro to Cells video, nearly all eukaryote cells have mitochondria. It's just that eukaryote cells that can do photosynthesis can have both mitochondria and chloroplast. Now, if you remember our beginning about theories, you may be wondering, where are the facts for this? We'll give you a few of the facts. First, mitochondria and chloroplasts have their own DNA. Yes, separate DNA from what is found in the nucleus. Not to mention that their DNA is arranged in a similar way to prokaryote DNA, specifically bacterial DNA. The size of mitochondria and chloroplasts tend to be similar to the size of bacteria. And when mitochondria and chloroplasts divide, which they can divide all on their own independently, they divide in a way that is similar to how bacteria divide. So, the endosymbiotic theory provides an explanation for how modern eukaryotes evolved from prokaryotes. There's obviously more questions you might want to explore. For example, what about some of the other structures and organelles in eukaryotes? Well, keep exploring. Secondary endosymbiosis is a great place to start. One last thing we want to make sure to emphasize. Endosymbiosis isn't just reserved for a theory that explains a past event in ancient history. No, because endosymbiosis is actually happening today with many other kinds of organisms. One of our favorite examples, the termite. Yes, termites can have prokaryotes that live in their gut and help them digest wood. And without them, well, let's just say digesting wood wouldn't happen so well. Well, that's it for the Meepa sisters, and we remind you to stay curious.
I hope you all have enjoyed the video. So next topic is the microbial epiphytes. So epiphytes, microbes, they are the group of the microbes that live on the surface of the plants. They can be found on the plant's leaves, stem, roots, fruits and seeds. So epiphytic microbe can be beneficial to the host or have a neutral or negative effect also. Plants present various microbial niches, collectively termed as a rhizosphere, that is the below ground, and phyllosphere, that is above ground, that is the leaf region. So this each niche hosts microbial, mutualist, commensals and the pathogens also, facilitating the range of microbe interaction, microbe-plant interactions. And these microbes and plants, they are important components of ecosystem, as we all know, and their interaction help in the regulating of the biogeochemical cycle in the environment. The microorganisms include bacteria, fungi, virus and some algae also, and they may be endophytic and epiphytic, that is based on the location on the host plants. And these microbes will use the host plant for their growth, colonization and proliferation, etc. And they also offer a variety of benefits to the host, including neutral or some of the negative effects also. Endophytic microbial interaction means that will influence the internal part of the plants, while epiphytic microbial interaction influence the exterior surface of the plant. This microbe also secrete the beneficial substance that will help in the plant growth, resistant to pathogenic microbes, removal of the harmful contaminants and production of secondary metabolites, and also contribute in the agricultural crop improvement, food safety and industries. Epiphytic microbes, to be more clear, they are those that live on the phyllosphere region. In this picture, we are seeing some of the examples of the epiphytic interaction. Here there are some of the microbial adaptation on the leaf surface. Particularly, I have taken example of a leaf here. So epiphyte must either tolerate the abiotic stress associated with the leaf surface or mitigate them through their own action. For this reason, successful epiphytic microbes, they must adopt a range of strategies to find or create a microhabitats that are protected from the abiotic stress, include manipulation of their host to modify the environment. So to survive the abiotic stress on the leaf surface, many microbes they form the biofilms. Biofilms, to be more clear, it is defined as an aggregation of the cell that is attached to a surface and embedded in an extracellular matrix. So these biofilms are actually resistant to the desiccation, antimicrobials and also ROS, that is reactive oxygen species, which are more important plant defenses. This can be actually formed even by the yeast and the filamental fungi also, and included a number of plant pathogens. Many epiphytic bacteria, they produce a plant hormones such as auxins, gibberellins and cytokinins in order to communicate and manipulate their host. For example, we have taken Pantoa agglomerans and the Pseudomonas syringae, as we are seeing in the pictures there. They produce an auxin, that is IAA, indole acetic acid. This actually causes the loosening of the plant cell walls, and they release the sugar, which is likely to increase the rate of the sugar exudation from the leaf. So, and also abscisic acid and the ethylene, they also affect the stomatal opening. They increase the water availability, while cytokinins, they trigger the release of the methanol, which is metabolized by some of the bacterial epiphytes. Another strategy that is followed by the epiphytic microbes to adapt on the leaf region is the secretion of enzymes such as cutinase, esterases, and lipases, so which can liberate nutrients from the cuticle by degrading the cuticular compounds. And phyllosphere microbes, they also have transporters such as ABC transporters, sporins, Toynbee dependent transport system. With this transporting system enhance the capacity for the nutrient uptake. Proteins of OMPA porine family, that is majorly found in the gram-negative bacteria. These are one of the most abundant that is found on the leaf surface. And these epiphytic microbes, they also produce the ion chelating siderophores to maximize the ion uptake. Some of the examples are pyocillins, pyoverdins, and pseudobactins from the Pseudomonas species. This siderophore production is also common among this epiphytic yeast, where it is associated with the biocontrol activities against the fungal pathogen. Next topic is the arrival of the microbes on the leaf surface and the assembly of the epiphytic communities. In the pictures we are seeing some numbers. These numbers represent the respective sentences as we are seeing that. The first one is the diverse microbes. There are different types of microbes as we are seeing in the number 1 that are landing on the leaves by various number of the roots. So that is by rain. Second number is rain, by the air movement. Fourth, from the leaf surfaces including the soil, leaves, rocks, and many fungal spores in the bacteria. And fifth, also they enter through the by the neighboring plant community composition and the seasonal factor. After arrival on the leaf surface, there is a formation of large aggregate of the microbes. So 7 number indicates once these on the leaf, these microbes exist in the large aggregates. The location and the size, they are determined by the heterogeneous nature of the leaf, surface topology, and the nutrient distribution. And 8th number tells that there is an aggregation of the aggregates that includes mixed species communities which may exist as a biofilms. I have already said you what are biofilms. Then 9th says that leaves, they harbor a less diverse microbiome than the surrounding environment, but partially due to the effect of the stress such as competition. And 10 says there is a high temperatures, UV radiation, and drying. 11th indicates low and heterogeneous water and nutrient availability. Where 12 says the presence of toxins such as plant defense compounds and the agricultural antimicrobials. So the influence of the host is then proposed to further select a specialized epiphytic microbiome. These are my references, and the video that I played was taken from the YouTube channel called as Amoeba Sisters. You can also visit the YouTube channel Amoeba Sister for more such beautiful and informative videos. I hope you have all got some information from this session, and also I have enjoyed this session. Thank you so much.